Hey guys, this is Mr. Razor. We are up to Chapter 12. Believe it or not, we are more than halfway done with the semester. I know it's been going pretty darn fast. I hope my visit with you last Friday was helpful. I hope I kind of cleared a few things up as far as how the grades were working and what you needed to do to be successful. I want you to know that I want to do everything that I can do to help you be successful. Of course, that is only a certain percentage of what's necessary. The largest percentage falls on you, the student, especially in an online course. That's a big responsibility, like I've mentioned before. But if you are doing your portion and you are studying and spending the time online and spending the time with your textbook and asking questions when necessary, I think you can be successful in this course, even though it moves pretty quickly and it can be somewhat difficult. Some of the subject matter can be difficult. So we are up to Chapter 12. We're going to be talking a little bit about reproduction. I don't want you to get too excited because it's just cellular reproduction. We're going to be talking about mitosis and the cell cycle. And this is a chapter that I think most of you will probably feel more comfortable with. This material I kind of felt more comfortable with. It's likely that you've seen this material more often, and so hopefully it'll be a bit of a break from chapters maybe 9 and 10 that were kind of hard on us. So chapter 12 is all about mitosis, and we'll get started in just one second. The chapter overview says the ability of an organism to produce more of itself or more of their own kind is one of the distinguishing characteristics that tells you that something is alive. It distinguishes living things from non-living things. Reproduction is a key characteristic or a key component of life. Rocks don't reproduce, plastic bottles don't reproduce, but living creatures, whether it's a single-celled amoeba or a tree or a dolphin or a human, they reproduce, they make more themselves. The continuity of life is based on this reproduction and something that we're going to look at called cell division, where cells come from other cells. Cell theory you've probably studied before. One component of cell theory is that cells come from pre-existing cells. And so there's a scientist who helped develop cell theory back in the 1800s, about 1855, Rudolf Virchow. He was actually a physician, and he was studying cells of animals and plants for that matter. And one of the things that he said was where a cell exists, it must have come, or there must have been a pre-existing cell. There was this sort of odd idea, or it sounds odd now, back then called spontaneous generation, where some people felt that living matter, living cells, living creatures could come from non-living material. For example, they thought that in garbage, perhaps somehow garbage created flies or maggots or created cockroaches, or bags of grain could create mice because they would see mice that would come out of a bag of grain when you would open a bag of corn, and they didn't really understand the idea of cells and reproduction all the way, so there was some thought that perhaps living material could spring from non-living. And so cell theory says cells come from pre-existing cells, that living objects come from other living objects. Now, cell theory does not address the first occurrence of life. So cell theory addresses the state of life as we know it now. So it says complex living cells come from other complex living cells through cell division. But that doesn't necessarily address the idea of how life first came into being. So I don't want you to look at cell theory and think, well, then how does that explain the originality of life, where life first began? Cell theory doesn't really address that. That's an entire another issue, and obviously an interesting one. Perhaps we'll have some time to get into that just a little bit as well. All right, so let's go ahead and continue. Great little picture here. This is the cell's chromosomes. So what they've done here is they've stained the chromosomes within the cell, which is going to be a major component of what we're looking at. And you can see a single cell chromosomes, and then they begin to form and map together to condense to where they're very visible. And you can see they have, in this picture here, can't really tell, but they have already duplicated, and then they line up, divide, and then here at the end you have two what we call daughter cells that are genetically identical. So this is kind of a cool poster or a cool picture that kind of shows, at least from the chromosome perspective, what's going on in cell division, and we're going to look at this in more detail. Cell division is important for all living creatures. It doesn't matter if you're made up of trillions of cells like a human being or you're a single-celled amoeba. A unicellular creature, when it divides, it reproduces an entire organism. So you start with one amoeba and you end up with two. But in a multicellular organism like a dog or a tree or a human, cell division is not going to reproduce an entire organism, but it is important for development of that organism. You started out, as all living creatures start out, as a single cell, a single fertilized egg cell, and now you're made up of trillions of cells. So there was a lot of cell division going on there. You are probably quite a bit larger now than you were on the day you were born. And that doesn't mean your cells have gotten bigger over the last 16, 17 years. It means the number of cells in your body have increased. So development, that is a big part of cell division. Growth, obviously, we just talked about how you have many, many, many more cells than the day you were born. And repair, if you get a cut or a scrape or you injure yourself, for that to be repaired, cells have to divide and make more of themselves. Cell division is a very integral, important part of something called the cell cycle, which is basically the life of a cell from its division, creation, throughout its entire life until it ultimately basically dies of old age. So one of the things we need to know is that this cell cycle and cell division is obviously important, but there's also some problems that can go wrong with it. And one of the good examples is various types of cancers. Cancer, we're going to look at toward the end of this chapter, is a good example of how this cell cycle can go wrong and certain things are not behaving the way they should. Your textbook always does a great job of showing various photographs, and so inside your textbook early in the chapter it shows these three photographs. At the top we have reproduction of a single-celled creature. So right here we have an amoeba basically splitting into two, and there'll be two whole separate amoebas from where there was one. Here we have growth and development. This is a sand dollar embryo. Perhaps you've seen sand dollars if you've ever gone to the beach, or you can buy them, basically the dried out shell of them at the store if you go to the beach or Galveston or Corpus Christi. So this is an embryo basically at the stage where the one fertilized egg cell has divided into two. And this would be like in a human body. This is tissue renewal, bone marrow. These are bone marrow cells that are dividing and creating more bone marrow. And inside the bone marrow is actually where new red blood cells are created. So three kind of different ideas or different purposes for cell division and this cell cycle that we're going to be talking about. Let's get started with concept 12.1. 12.1 says most cell division results in genetically identical daughter cells. The type of cell division we're going to be looking at in this chapter is called mitosis. That is where the cell divides and it creates a clone of itself. And this is what most cell division is all about. We're going to call them daughter cells, even though obviously we're not talking about a male or female here, but they're called daughter cells, and they have identical genetic information, identical DNA. So they're basically a clone. So when one of your skin cells divides into two to, say, repair a cut, that skin cell is going to be identical. Those two skin cells are going to be identical to each other, basically clones. There's another type of cell division we'll talk about actually more in Chapter 13 called meiosis, sometimes pronounced meiosis, and I'll tell you why in just a moment. But meiosis is a special type of division that produces the sex cells, the gametes, which would be sperm or egg cells. They are different, and we'll find out how those are different in the next chapter. When I was in middle school, one of the ways I tried to remember mitosis from meiosis was I would think, okay, mitosis was how my toes were created, were basically make more of themselves. As my toes grow, those skin cells and muscle cells divide. And so they're creating clones of each other. So when body parts grow or body parts repair, that's mitosis. And then the other word is meiosis, but as a kid I would pronounce it meiosis to remind myself that this is what was required to create me. When my parents reproduced, I was a combination of genetics from my mother and father, and that was a different type of cell division that created the sperm and egg. So meiosis was required to produce me. Mitosis is what goes on inside my toes. So it's very silly, but I think I have remembered that all the way back from 7th grade. So do whatever you can, make up whatever silly rhyme or silly saying you can to try to remember some of these vocabulary words. That's what did it for me. You choose what will do it for you. Now when these cells go through mitosis, when they divide, they're basically doubling up everything inside of them and then dividing in half. So all of the cytoplasm, they create more cytoplasm, they create and enlarge their plasma membrane, they create extra copies of the Golgi apparatus and the mitochondria, and basically they replicate or double everything. But perhaps the most important thing that they replicate is the actual genetic material, the DNA. This is called the genome. All of the DNA inside the cell that basically is the blueprint for that cell is called the cell's genome. And a genome can be fairly small. It can be a single strand of DNA, a single DNA molecule, like in some of the more simple prokaryotic cells, or it can be more complex. It can be a large number of strands of DNA. Say, for example, in most eukaryotic cells, humans and dogs, mammals, birds, that's going to be a larger number of strands of DNA or molecules of DNA that make up the genome. You may have heard in the news recently, there's been a lot of talk about genome sequencing, where we have basically kind of broken the code of a particular creature's genome, and that was done with humans probably a few years back now, not too long ago, and we have done that with the genome of a wide variety of living creatures now, thanks to the help of powerful computers and new technology. The DNA molecules that are within the nucleus, within the cell, are basically packaged in something that we call chromosomes. So that thin strand of DNA is going to be called a chromosome, and different types of species of animals and living creatures have different numbers of chromosomes. Each eukaryotic chromosome is made up of one linear strand of DNA wrapped around a number of proteins. And we'll see a picture of that coming up. And sections of that strand are what we call a gene. So you have a chromosome, which is really just a long strand of DNA, and certain chunks of that strand or sections of that strand are called a gene. And that section corresponds to a particular characteristic. So maybe this one particular section controls eye color, or this one particular section controls whether or not you can curl your tongue into a taco shape. So it's more complicated than that, generally speaking, but in some cases only one portion or one gene controls the trait, and so that's what a gene is, a portion of the DNA that controls a particular trait. A typical human cell has a pretty amazing amount of DNA actually inside. If you were to stretch it out and lay it out end to end, of course it would be not visible to the eye without a microscope, but it would actually measure over 2 meters long. And it's kind of crazy to think that all of that has to be copied and replicated exactly and then divided into two to make the 2 daughter cells. Here's a great shot of a eukaryotic cell showing kind of staining the chromosomes within the nucleus. So the dark areas that you see here are the chromosomes, not always visible under a light microscope, but once they start to condense in preparation for division, oftentimes they can be seen underneath a light microscope. So all of this mass of strands of DNA that you see inside here, those would be the chromosomes of this eukaryotic cell. For the most part, we're going to be focusing on the eukaryotic cells. So eukaryotic chromosomes, I already said, consist of strands of DNA, long filaments or strands of DNA. We call this chromatin, and that would be the strand of DNA wrapped around the various proteins that make it up. So think about, like, say for example, if you had a long piece of string and you had that string laid out on the table, but at certain points that string was kind of wrapped around a little stone, and then it kind of trailed off and it was wrapped around another stone, and then it trailed off farther. That's kind of what it is like, or kind of what it looks like. This chromatin is basically the strand of DNA wrapped around and kind of in conjunction with various proteins. This is usually fairly long and slender, but we're going to find out that it kind of condenses and squishes down before cell division. Every eukaryotic species, whether you're talking about a bird or a fish or a mammal, a human or a dog, they have a characteristic number of chromosomes in the cell nucleus, and that number can vary pretty widely. So the number of chromosomes within what we call somatic cells, which are the non-reproductive cells, that would be like, say, skin cells or liver cells or muscle cells. The number of chromosomes within the somatic cells can vary quite a bit. So, like for example, in some species of cabbage, there's about 18 chromosomes within the cell. In chimpanzees, it's 48 chromosomes. In humans, it's 46. In hedgehogs, it's 90. And there's even one species of algae, which you would think would be a very simple type of living creature, that has 148 chromosomes. So the number of chromosomes doesn't necessarily correspond with the complexity of the organism. We have two different types of cells in the body. You see somatic cells here. Somatic cells are all of your body cells that are not the reproductive cells. So your skin cells, your liver cells, your muscle cells, your cells of your digestive system, those are all called somatic cells. In humans, there's 2 sets of chromosomes in those cells, 23 pair. So there's 23 pair of chromosomes for a total of 46. There is one other kind or type of cell within the body, and these are called gametes, or reproductive cells, or sex cells. That would be the sperm, of course, in males and the egg in females. These are slightly different. We're going to find out in the next chapter that they only have half as many chromosomes as a normal somatic cell. And that's probably obvious because those two are going to fuse together to form the new creature, what eventually would become the baby of that particular living creature. So somatic cells are the cells that are non-reproductive. They have the full set of chromosomes. Again, in humans, that's 46 chromosomes, 23 pair. We're going to find out in the next chapter half of those pairs, half of that of each pair came from mom, and half of each pair came from dad. So we have somatic cells, and then gametes are the specialized cells, the reproductive cells, that we'll probably more take a look at those more in the next chapter when we talk about meiosis or meiosis. So as these cells get ready to replicate or divide, they double up pretty much on everything inside, and then they begin to replicate the DNA, probably the most important part. So they basically have to make a photocopy of this DNA, and it needs to be an exact copy. If there's any error, that can be problematic. That can cause a mutation. Not all mutations are negative. Some actually turn out to be helpful or positive, but preferably we would want an exact copy. So the DNA replicates or is copied, and then it begins to condense, and the chromosomes basically will curl up on themselves and condense down in size. Each duplicated chromosome has what we call two sister chromatids, and I'll show a picture in just a moment. A chromatid is basically the two copies of that chromosome joined together sort of at the waist, and you've probably seen this before. Those will eventually separate when the cell divides, but for the moment they are joined together sort of at the waist or at the hip at a small area called the centromere. The centromere is that narrowed down area that kind of looks like a waist or the center portion of the X of the chromosome. So that's where the two sister chromatids are most closely attached, sort of in the middle there. So these are normally, the DNA is normally in long, thin fibers, but after it divides, after it replicates, it begins to condense, and these condensed versions oftentimes are fairly easily viewed under a light microscope, certainly viewable under an electron scanning microscope or something similar. So here's a great picture of two sister chromatids joined together at the centromere. Now, one of the problems with the vocabulary is the single strand of DNA within the cell is a chromosome. But oftentimes, what you see in this picture is called a chromosome 2. Well, this is actually two sister chromatids, which is a double copy of the chromosome. So it can get kind of confusing because sometimes the single strand is called a chromosome, and oftentimes this doubled up strand that's connected here is called a chromosome also. So you want to be careful about what you're calling a chromosome. This would probably be better off calling it a pair of sister chromatids as opposed to a chromosome itself, but you see that happen very often. So this is a highly condensed, duplicated human chromosome made up of two sister chromatids. This chromosome up here is an exact copy of the chromosome down here, and they are joined together pretty much throughout this entire area, but most closely right here at this portion called the centromere. During the process of cell division, eventually these two sister chromatids that we just saw, they're going to be pulled apart, separated, and they will move towards the two opposite ends of the dividing cell. And so basically we're going to double up on the DNA material, then we're going to split those doubles apart, and then separate them into the two new cells. And then once they separate, those chromatids, so sister chromatids, are single strands again called chromosomes. And again, like I said, unfortunately the word chromosome is often used to describe the single strand as well as the joined two sister chromatids. So that can cause some confusion. Here's a great little illustration or drawing that shows a very simplified version of the process. So here we have a chromosome. We're just looking at a cell that has a single chromosome, just to make it simple. So here we have a chromosome. The centromere is that little wasted area, the thinned out area toward the very middle here. In actuality, in the beginning, the chromosome wouldn't be all lumped together like this. It would be long and thin, but they're showing it condensed just to make it more clear. That chromosome is basically made up of a single DNA molecule, a single strand of DNA. Then the chromosome duplicates, and we have two sister chromatids connected together at the centromere. Oftentimes looks like a little X. It's made up of two duplicate copies of the DNA molecule. And then the last step is to separate, and one of the chromatids goes one way, one goes the other, and then we have basically duplicate copies of each other. Now, I wish it could stay that simple, but of course we're going to go into more detail. So in eukaryotic cells, we call this mitosis. And usually when I say the word mitosis, I mean the entire process of a skin cell dividing into two skin cells. But in actuality, it's kind of two different processes. Man, that's hard to say. Mitosis is the division of the genetic material. So the division and copying of the genetic material is the actual mitosis. And then the splitting of the cell, the basically dividing into two pieces and separating, is called cytokinesis. That's cyto meaning cytoplasm. Kinesis is movement. So cytokinesis is the division of the cytoplasm. So oftentimes mitosis, that word covers everything. But if you wanted to be specific, you're going to have mitosis, which is division of the DNA and replication of the DNA, and then cytokinesis, which is division of the actual cell itself, the cytoplasm and the cell membrane. The gametes, we talked about a little bit earlier. Those are a slightly different kind of cell that we're going to look at in the next chapter. The gametes are produced by a slightly different method. That method is called meiosis, and again, we'll look at that in the next chapter. Meiosis does not give you two exact copies. Meiosis does not give you a clone like mitosis does. Meiosis yields non-identical daughter cells, and these are sex cells or gametes, and they only have a single set of chromosomes, half as many as the normal cell, half as many as a somatic cell, half as many as the parent cell that they came from. But we're going to look at chapter 13 for more of that, and pretty much for the rest of this chapter and the next, we'll kind of keep our eye on eukaryotic cells. We will talk about some of the other kinds a little bit, but eukaryotic cells will be our main focus. All right, before we get into all the steps of mitosis, let's take a quick break, and when we come back, we'll get into concept 12.2, and that'll be the actual steps of mitosis, and we'll get into a bit more detail. See you back in just a minute.